Asymmetric PCR. Asymmetric PCR is used to amplify one strand of the original DNA more than the other strand. In this type of the PCR, because the DNA is double-stranded, during this type of the PCR, one strand is more amplified as compared to the other strand. It is used in some type of the sequencing and hybridization probing where having only one of the two strands is needed in more quantity. In case of asymmetric PCR, PCR is carried out as usual or as a normal PCR but with great excess of one of the primer. One of the primer is in large quantity while the other primer is in less quantity. Due to slow amplification in the later stages due to less amount of one of the primer, that is also called as limiting primer, extra cycles of the PCR are required. Mostly in case of asymmetric PCR, 50 to 55 cycles are performed for amplification with the help of one limiting primer and one normal amount of the primer. It finds use in some of the sequencing and hybridization probing where only one of the two complementary strands are required. So if we conclude that asymmetric PCR is such a PCR in which there are one primer in excess amount and the other primer is in less amount. So by using this kind of the primer amounts, we can amplify one of the strand in large amount. We can produce more copies of that strand, and while the strand for which we have used less amount of the primers, there will be a very less number of copies produced for that strand. This type of the PCR is called as asymmetric PCR.